You can have perfect hormone levels, flawless baseline tests, be incredibly healthy, and still sit in a clinic just watching your IVF cycles fail.
Yeah, it's heartbreaking.
It really is. You produce embryos, but they consistently grade out as poor quality. It's a devastating reality for so many couples, and honestly, it highlights this massive biological blind spot.
Right, because we can sequence a genome, we can measure hormones down to the picogram, but we still struggle with this one fundamental question.
Which is, why do some microscopic cells thrive and become, the foundation of a life, while others that look perfectly identical on paper just falter?
And that is really the absolute frontier of reproductive biology right now. I mean, we are dealing with microscopic margins of error here.
Yeah, like razor thin.
Exactly. A slight imbalance at the cellular level doesn't just cause a little hiccup, it completely arrests the development of life. Genomics can tell you what a cell is capable of doing, right? And proteomics tells you what machinery it has already built.
But neither of those tells you what's actually happening in that exact moment.
Spot on. They miss the real-time activity.
Okay, let's unpack this. We're taking a deep dive into a really groundbreaking 2026 paper from Cell Death and Disease. Our mission today is to uncover the hidden metabolic fingerprint of oocyte, that is, egg quality and embryonic development.
It's a fantastic paper.
But before we get deep into the cellular mechanics, I'm genuinely stuck on the logistics here. How are we predicting the viability of a microscopic embryo before it's even fully formed? Especially since we obviously cannot, cut it open to look inside.
What's fascinating here is that we look at the exhaust, we look at the fuel, we look at the metabolism.
The metabolism.
Yeah. Metabolism is the ultimate endpoint of biological regulation. It is the real-time, immediate reality of a living cell.
So it's like if you want to know how an engine is running, you don't just read the manufacturing blueprint.
Exactly. You check the temperature, the fuel consumption, the exhaust. By analyzing the metabolic environment surrounding the egg, we get a highly accurate picture of the physical trait, the phenotype, without ever disturbing the embryo itself.
And the molecule at the dead center of this metabolic reality, the one we're going to be talking about extensively today, is adenosine. So keep that name front and center.
Oh yeah, adenosine is the absolute star of the show.
To understand why adenosine is the key, we have to look at the clinical evidence. The researchers didn't just start in a petri dish, right? They started with a cohort of 36 couples undergoing IVF.
And the data here paints a very specific picture.
Right. Let me read some of these stats because they are wild. The female partners had a mean age of 33.3 years and a super healthy BMI of 22.6. Their ovarian reserve tests were rock solid. Basal FSH levels were at 7.6 IU per mil.
Which is totally normal.
Yeah, and their AMH, the atomullarian hormone, which tells us about egg supply, was at 4.3 nanograms per mil.
Those numbers are critical because they rule out the usual suspects. I mean, these are not patients in severe reproductive decline.
And the male partners were equally healthy, around 35.5 years old, normal sperm density at 59.5 million per milliliter.
A really solid count.
And motility was at nearly 55%. When they actually went through the retrieval process, the clinics got an average of 9.6 oocytes per cycle, with a very standard 71.2% fertilization rate.
So statistically speaking, these 36 couples are practically perfectly matched across all the clinical factors.
Right, but the outcomes completely diverge. The researchers split them based on a rigid threshold. Did the cycle produce less than 40% high-quality grade 1 and 2 embryos, or did it produce 40% and above?
This is where we have to sort of visualize the microenvironment. The oocyte doesn't just exist in a vacuum.
No, it's got a whole support system.
Exactly. It sits at the center of a highly complex network, so directly surrounding the oocyte are the cumulus cells. Think of them as the power stations and the supply lines.
Power stations, I like that.
Yeah, they filter nutrients, they provide energy, and they send critical molecular signals directly to the egg. And then surrounding all of this is the follicular fluid.
Which is like the moat around a castle.
A moat, or maybe more like a regional transport highway. It carries hormones and metabolites to and from the entire structure.
Okay, power stations and highways, that works better than the old queen in her castle analogy because this is an active dynamic system. So the researchers couldn't touch the oocyte itself, so they analyzed the power stations, the cumulus cells.
Right. And to do that, they used a technique called UHPLC-HRMS.
Which is quite the mouthful. That's ultra-high-performance liquid chromatography coupled with high-resolution mass spectrometry.
Basically a massive machine that weighs and identifies every single microscopic molecule in a sample.
And they found 148 different metabolites in there. but only 15 of them were significantly different between the high-quality and poor-quality embryo group.
And here is the kicker. Out of those 15, 14 were abnormally high in the poor quality group.
Wait, higher, not lower.
Higher. They were just accumulating in the cumulus cells. When the researchers ran a pathway enrichment analysis, which is just a way to see what biological system these molecules actually belong to, they found a massive disruption in purine metabolism.
And the primary culprit in that pathway was adenosine.
Yes. In the cycles that produced poor embryos, adenosine was severely trapped inside the cumulus cells. The predictive value of this, measured by a stat called the ROC-AUC, was 0.7465.
Which, in the world of biomarkers, that's a pretty robust, reliable signal, right? Like a solid B grade for predicting outcomes.
Exactly. It's a localized metabolic traffic jam. The adenosine is just stuck in the power station.
And when they checked the regional highway, that follicular fluid, they found it was practically empty. The fluid surrounding the poor quality eggs was drastically depleted of adenosine.
The fuel just isn't circulating. It is being hoarded inside the support cells, leaving the surrounding environment starved and the entire metabolic network compromised.
Let's synthesize this for a second, just to recap before we move on. Number one, baseline clinical stats like age and AMH don't tell the whole story.
Right, there's a hidden metabolic layer.
Number two, cumulus cells, oocytes, and follicular fluid form this codependent microenvironment. And number three, poor embryo quality features this massive adenosine imbalance. Too much trapped inside the cells, not enough in the fluid.
Which raises this immediate biological question: how does a cell actually become starved of its own fuel in the middle of a nutrient-rich environment?
Right. And I actually want to throw a question to you, the listener, right now. Think about your own daily energy levels. If the fuel gets trapped inside one room of your house and can't circulate, what happens to the rest of the system? What stands out to you about this cellular traffic jam?
It's like having a full fridge, but the kitchen door is locked.
Exactly. And that is a perfect transition into how we grade the evidence here. Clinical observation in humans is great. It gives us correlation. But to prove causation, to prove this trapped adenosine is actually causing the poor embryo quality we have to move to animal models.
We have to manipulate the variables.
So the researchers didn't just use one model, they used two independent models to simulate declining oocyte quality in 7-week-old female mice.
Right, the ICR mice.
Yeah. One group was subjected to whole-body X-ray irradiation at 0.5, 1, or 2 grays, and the other group was injected intraperitoneally with cyclophosphamide, or CTX, which is a chemotherapy drug, at 75, 100, or 150 milligrams per kilogram.
Now, why use both radiation and chemo? Because they induce cellular stress and diminish ovarian capacity through entirely different biological mechanisms.
Oh, I see.
Yeah, radiation causes direct physical damage to cellular DNA, while CTX chemically disrupts cell division and induces toxicity.
So if both of these vastly different stressors result in the exact same adenosine trap, it proves that this metabolic bottleneck isn't just a quirk of radiation, right?
Exactly. It proves it's a universal, fundamental biological response to declining ovarian health.
And it absolutely was. After 1 week, both the irradiated mice and the CTX-injected mice showed severely decreased ovarian size and a plummeted follicular index.
And when they ran that targeted metabolomic analysis again, the mice showed the exact same traffic jam as the human women.
Adenosine hoarded in the cumulus cells, totally missing from the follicular fluid. But, knowing it happens isn't enough. I want to know the why. Why is the adenosine getting trapped?
Right. Who built the roadblock?
Yes. To find the roadblock, they analyzed the cell's physical infrastructure using quantitative RT-PCR.
Or QRT-PCR.
Right. This technique basically acts like a molecular tally counter. It counts the exact number of messenger RNA molecules being produced, telling us precisely which genes are turned on and which are shut down.
And they discovered a catastrophic collapse in the expression of specific transport genes.
The locked doors and broken bridges analogy.
Precisely. Let's start with the doors. The researchers found a massive decrease in ENT1 and ENT2.
Which stands for equilibrium nucleoside transporters.
Right. A good trick to remember this is ENT equals entrance, or exit, really. These are the physical doors embedded in the cell membrane that allow adenosine to flow out of the cell and into the follicular fluid highway.
So in the low-quality oocytes, the expression of ENT1 and ENT2 was severely downregulated. The doors were quite literally locked and bolted.
But they also found a breakdown internally. It wasn't just the doors to the outside highway.
Right. They looked at the internal intercom system, too.
Yeah, they found a sharp decrease in a gene called CX37, which builds a protein called connexin 37.
Remember, CX equals connection.
Yes, CX37 forms what we call gap junctions. If ENT proteins are the doors to the outside world, gap junctions are the internal microscopic bridges directly connecting the cumulus cell power stations to the oocyte queen.
They allow molecules to pass back and forth instantaneously.
But when CX37 shuts down, those bridges collapse.
Here's where it gets really interesting. If we go back to our power station analogy, the ENT doors opening to the highway are locked shut, meaning no fuel gets to the broader region. And the CX37 tunnels connecting the power station directly to the central queen are collapsed.
The cell is completely isolated. The cumulus cells just drown in a massive accumulation of unused adenosine, while the oocyte and the follicular fluid outside just starve.
And in biology, isolation equals stress. When an oocyte loses its communication channels and its extracellular signaling molecules, its internal regulatory networks begin to just unravel.
It is entirely walled off.
So recap time for subtopic 2. 1. Mouse models using X-ray and CTX confirm the human findings of diminished ovarian capacity. 2. Adenosine accumulation is caused by the downregulation of ENT1 and ENT2 transporters.
Locked doors.
Right. And 3. Gap junction communication breaks down due to reduced CX37, the broken intercom.
It's a devastating structural cascade.
So listener, think about this. If the doors are locked and the intercom is broken, how does a cell signal for help? Have you ever experienced a breakdown in communication that just cascaded into a bigger catastrophic failure?
Like when one server goes down and the whole company network crashes?
Exactly. And that is what's destroying these early embryos. Which brings us to the intervention phase of the study. The researchers asked a beautifully elegant question. If the doors are locked from the inside, what happens if we just bypass the infrastructure entirely and deliver the fuel from the outside?
The rescue mission.
Right. They took these aged, low-quality, stressed oocytes and placed them in an in vitro culture medium. But they altered the medium by simply adding exogenous adenosine, manually replacing what the fluid was missing.
And they only cultured them for 12 hours.
Which is wild. Just 12 hours of exposure to external adenosine, and the results were stunning.
It was nothing short of a total cellular reboot. The rates of embryos reaching the 2-cell, 4-cell, and blastocyst stages just skyrocketed.
He completely restored the developmental potential.
Furthermore, the quality of these blastocysts improved dramatically. They had way higher total cell counts and a severe reduction in apoptotic or dying nuclei.
Okay, wait, I really have to push back here because biology is rarely this simple. You were telling me that these cells had radiation damage, chemotherapy toxicity, locked transport doors, and collapsed internal tunnels.
Yep.
And just floating them in a bath of a single molecule adenosine for half a day fixed that catastrophic structural damage.
Yeah.
How does the cell even know what to do with it if the doors are locked? Walk me through the algorithm.
It's a fantastic question. The key is that the cell doesn't need the doors to be open to receive a signal from the outside.
Oh, because of receptors?
Exactly. The researchers used a technique called SmartSeq2 transcriptomics to read the cell's real-time genetic instruction manual during this rescue. And they found that when you add exogenous adenosine to the fluid, it binds to adenosine receptors, ARs, which sit on the outside surface of the cell membrane.
So think of these AR receptors like satellite dishes on the roof.
Perfect analogy. When adenosine docks into the dish, it triggers a massive, complex intracellular cascade. It physically rewires the cell's behavior from the outside in.
And the data showed 2 distinct pathways firing up to save the cell. Let's break down the molecular algorithm. Pathway 1 is all about meiosis and spindles.
Right. So when the adenosine receptor is activated, it triggers a second messenger inside the cell called cyclic AMP, or CMP.
That's the runner.
Yes, the CMP acts as a runner, carrying the signal deep into the cell to activate an enzyme called pKa. This CAMP-PKA pathway is the master regulator for cell division.
Because an embryo's very first job is to divide safely. 1 cell becomes 2, 2 become 4.
Exactly. And to divide safely, the cell has to perfectly align its chromosomes and pull them apart using a physical scaffolding called the meiotic spindle.
Okay, so how does pKa control the scaffolding?
The CAMP-PKA pathway directly regulates 2 massive protein complexes. The first is MPF, specifically a protein called Cyc-B1, which drives the division forward.
Like the gas pedal.
Right. And the second is APCC, specifically CDC27, which acts as the checkpoint to ensure everything is aligned.
The
Yes. By activating this pathway, the exogenous adenosine acts like a master conductor, fine-tuning the gas and the brakes so that the chromosomes don't tangle, preventing meiotic errors.
Wow. So pathway 1 is essentially rebuilding the physical scaffolding of the cell so it doesn't tear its own DNA apart when it divides. That is incredible. But what about pathway 2?
Pathway 2 deals with the chemical fire of oxidative stress.
The rust.
Exactly. When the cells were originally starved of extracellular adenosine, it triggered a massive panic response. The cell upregulated a gene called MAP8, which produces a highly inflammatory protein pathway known as JNK.
And high levels of JNK basically mean the cell's engine is redlining, producing toxic exhaust.
We call that exhaust reactive oxygen species, or ROS. ROS are these highly unstable molecules that steal electrons from other cellular structures. They literally rust the internal machinery of the cell.
And the researchers proved this using fluorescence staining, right?
Yeah, they used CM-H2D-CFDA and DHE fluorescence staining. These dyes physically glow bright red or green in the presence of ROS, and the low-quality stressed eggs were glowing incredibly brightly under the microscope.
But when they introduced the exogenous adenosine to the satellite dishes on the cell membrane, the internal algorithm shifted.
The adenosine signaling actively suppressed the MATCake8 gene and shut down the JNK pathway. The fluorescent glowing faded, the ROS levels plummeted.
It extinguished the fire. So let's recap subtopic 3. 1, exogenous adenosine supplementation rescues early embryonic development.
Huge finding.
2, it fixes meiotic defects via that AR to CAMP and PKA pathway, regulating the CycB1 and CDC27 scaffolding.
The gas and the brakes.
And 3, it mitigates oxidative stress by suppressing the JNK pathway, slashing the levels of toxic ROS.
It is a profound demonstration of how a targeted metabolic intervention can just override deep structural cellular stress.
It really is. So listener, think about this. We often think of stress as a mental burden, right? A tough week at work. But here we see oxidative stress literally breaking cellular spindles apart. How might optimizing our own cellular metabolism protect us from systemic stress?
It really forces you to view your health at a completely molecular level.
It definitely does. So what does this all mean? We started with couples sitting in fertility clinics, desperate for answers as to why perfectly good baseline stats lead to failing embryos.
If we connect this to the bigger picture, the clinical implications here are going to totally shift the paradigm of IVF.
Because it's not just a new additive for the Petri dish, right?
Well, yes, obviously this points toward a highly effective additive for in vitro maturation mediums. Clinics could potentially rescue struggling oocytes simply by optimizing adenosine in the culture fluid. But the bigger breakthrough is diagnostic.
The biomarker aspect.
Yes. This study establishes adenosine disequilibrium as a highly predictable, mathematically measurable metabolic fingerprint.
It removes all the guesswork. I mean, historically, embryologists have had to rely on subjective visual grading. They just look at an embryo under a microscope and make a judgment call.
Right. They say, oh, this looks like a great eye, this looks a bit fragmented. But visual grading tells you the damage has already happened.
Too late.
Exactly. This research turns that subjective art into an objective, precise metabolic science. By analyzing the fluid from the cumulus cells during retrieval, clinicians can measure the exact intra versus extracellular adenosine ratio.
They can read the metabolic diagnostics and predict the viability of the embryo before the visual defects ever even manifest.
It's the difference between waiting for your car's engine to seize up in smoke versus plugging a computer into the dashboard and seeing that the fuel pump is about to fail.
And intervening before the failure happens.
Exactly.
As we wrap up, I want to leave you with one final lingering concept to really ponder. Throughout this entire deep dive, we've talked about adenosine as the crucial fuel for an embryo. But adenosine isn't just an obscure reproductive chemical.
No, not at all.
It is the A in ATP, adenosine triphosphate. It is the absolute universal energy currency of all living things.
You cannot move a single muscle or fire a single neuron without it. It's the backbone of life.
So if a microscopic imbalance of adenosine, just a tiny trafficking error across a few locked cellular doors, can dictate the very dawn of human life in an embryo, what role might it play in the broader aging of our adult bodies?
Wow. If broken bridges and locked transport genes cause this much chaotic stress in a single cell, imagine the cumulative effect across trillions of cells in a 60 or 80-year-old biological system.
Right. Could the onset of age-related diseases just be a macroscopic version of this exact adenosine trap? As we get older, our cells just slowly locking their ENT transport doors and shutting down their CX37 communication tunnels?
Could mastering adenosine transport be a key to anti-aging itself?
Exactly. It makes you wonder. Thank you so much for joining us on this deep dive. Keep questioning the microscopic mechanisms that make life possible, and keep looking for those hidden metabolic fingerprints. We'll catch you next time.